What is up, boys and girls and everyone else? This is another science video lesson episode right here, and for this episode we will be going to talk about two important processes in the cell necessary for the growth and development of an organism. Come join me today as we discover how DNA replication and protein synthesis works as part of the characteristics of life called growth and development. Alright, so here we are in our topic for this video lesson. So before we go to our main topic, so let's take a look first on the things that you see on this screen. So the things that you see on your screen right now is dots and dashes, okay? And these dots and dashes that you have right here correspond to a certain letter, okay? Like what we have right here, we have dots and dash that corresponds to letter A. Then we have single dash and triple dots that you have over here that corresponds to letter B. Now, if you combine them all together, you will create a phrase or even a sentence. Alright, so this is the Morse code. Alright, so the Morse code is one of the way of communication during the 1900s. Even the Titanic, before it sank in the bottom of the ocean, it uses a Morse code. It uses a Morse code in order to communicate to other ships around it. Okay, so that is the Morse code, and the Morse code was used in the discovery of the relationship between electricity and magnetism way back. It was discovered by Michael Faraday. So people during that time make use of this concept and created this new way of communication. So basically, this is the ancestor of the modern smartphones that we are using right now. This Morse code that we have right here. Now, okay, you need someone who can interpret these codes that you have right here. So you need an expert person or a trained person who can interpret this combination of dots and dashes that you have over here. So during that time, the operator should be experienced enough in order to decode the message using the Morse code that we have right here. So to show you how it works, so I went to a website that generates a Morse code, and I have it like this. Okay, so you can go to this website, or even you can type a Morse code generator on the Google search, and there are a lot of results there. You can try everything there. Alright, so what the one that I have right here is the mobilefish.com Morse code generator. So you can look at it in the description below this video. So what is on this website? So it generates Morse code. Okay, it generates a Morse code, and from your message, it will transform that message to become a Morse code. Alright, so okay, if you are expecting that that's how it is easy to do, so it is not. Okay, during the 1900s, it is very a complicated process. Okay, so for example, if you will send an SOS message using a Morse code, so first you need to find out what are the corresponding codes that corresponds to the letter S, O, and another S. Okay, so if I'm going to play this one, so it sounds like that. Alright, so I don't know how the transition works or how the spaces in this particular message works, but it is utterly amazing. Okay, if you will hear this one. So let's play this one once again. Alright, so that was the SOS message. Okay, and in this website, you can create a you can have this message. For example, I have this okay, so I can type here is message. Okay, where am I today? Right. So, for example, I have this message that we have right here, and just type there the access code, then it will create the Morse code at the bottom. Okay, so if you were going to play the message that you have right here, Alright, so that was the sound, okay, of this message that I have created over here. Okay, so it is an interesting one, but the thing there is during those times in 1900s, it is somehow complicated. Okay, even the transcription, all right, or even the translation of these codes that you have right here, it is super complicated. Okay, so it's super complicated. So you will admire those people who are very experienced enough to translate this particular message into from Morse code to a message, or message to a Morse code. Okay, so that was the Morse code. All right, so okay. So probably if all of our digital devices suddenly went down, probably we will go back again to this old way of messaging. Okay, the Morse code. Okay, so another form of messaging is writing, something like that. Okay, so if you happen to heard before the Dead Sea Scroll, okay, so it was found in the Dead Sea. Okay, so these are scrolls. These are artifacts that were found in the Dead Sea, and it is written, or what is written on the Dead Scroll are Hebrew languages, Hebrew phrases that you have right here, and you need someone who is experienced enough to read and understand what the Hebrew writings is all about. Okay, so fortunately enough, there are a lot of experts about this type of language that you have right here, and they be able to translate. Alright, they be able to translate what is written here in this Dead Sea Scroll, and this is part of our Bible right now. Okay, this is part of the Bible. So the Old and New Testament of the Bible was based on the writings, was based on the artifacts that was found in the Dead Sea Scroll. Alright, so basically all the things that you found in the Bible came from these artifacts. Okay, so since we are talking about something that is interpreted, when you interpret it, you translate it, and you translate it to something that is more understandable. Alright, so the reason why we can understand this language that you have right here, it's because we can translate it in our own language, which is the English language. Alright, and the Morse code that you have right here, from dots and dashes that you have, we can translate this one into a more language that we can understand, and that is the letters that you have over here, the numbers, and then the punctuation marks. Alright, without these punctuation marks, the numbers, and then the letters that we have over here, probably we will not be able to understand what this message is all about. Alright, so since we are talking about that one, the topic on this video lesson has something to do on that. Alright, so but before we go to that, let me give you first some words that we might encounter in the long run when we are discussing this particular topic. So we have here the word polymerase, which means synthesize. Alright, so polymerase, which means synthesize. And we have here the helicase, means bind, or sometimes it is called remodeled. Right? So helicase is the synonymous word for helicase is remodeling. All right. Okay. Next one, we have this ligase. Ligase means joining. All right. So joining. So these are some words that we might encounter as we discuss this particular topic. So all of them are enzymes. All right. All of them are enzymes that are needed in this topic that we have right now, which is called the protein synthesis. So this is the process on how the DNA makes protein. Okay? So but before we go to the process itself, we will start first with the more basic idea. Okay? Before going to the protein synthesis process itself. Alright? So we'll start first with the idea of what they call the DNA replication. Okay? So the DNA replication process. So okay, are you ready, guys? So let's go on this topic that we have right here. What is DNA replication, and what is the importance of the DNA replication in our body? Alright, so what is DNA replication? So what is DNA replication? This is the process by which the DNA makes its own copy during the cell division. Alright, and this particular process happens during the S phase of cell division. So when you were grade 8, So when you were grade 8, you have encountered this phases of cell division, and this is where the DNA replication happens. Okay, so in the process of cell division, especially the mitosis, there are several phases. So it starts first with the longest phase on the cell division, called the interphase. Alright? Followed by the prophase, metaphase, anaphase, telophase. Alright? So now to start with, in the cell, before cell division, it starts first with the G1 phase. Alright? So the G1 phase of the interphase, what it does is it prepares the cell for duplication. Alright? It prepares all the contents of your cell for duplication. Alright? And when it undergoes now to the synthesis phase, or the S phase that you have over here, so the duplication process or the replication process will now take place. Okay? Then after that one, after the replication process, we have now the G2 phase. That's the last portion of the interface that we have right here. Now, what it does, or what happens during the G2 phase, or G2, in short, what happens during that phase is the DNA, the cell proofreads the DNA that was created. Alright, so it proofreads the DNA that was created. And what is the reason why it proofreads the DNA? It's because it wants to find out, or the cell wants to find out, if there is something wrong after the replication process. Okay, so if there's something wrong, the DNA will be discarded. And if there is something unusual on the sequence of the DNA that was produced, okay, it will repair that particular errors that was proofreaded. Okay, so that was the G2 phase. So all of this thing that you have right here, from G1 to G2 phase, are part of your interface. Alright, now after the interface, we have now the prophase, then followed by the metaphase, then followed by anaphase and telophase. So it happens during the mitosis. Okay, so it happens during the mitosis, and S phase is one of the most important part in this process. Because without the S phase, the DNA will not be able to, or the cells, instead of containing the same genetic information, the cells will not have any genetic information at all. Okay, so without this S phase. All right, so how is the DNA replication works? All right, so after its discovery way back in the 1900s, all right, so 1940s, something like that. All right, so after its discovery, okay, so scientists or biologists do not have any idea how it replicates. Okay, so you know the people who discovered the DNA. So it was Francis Crick and James Watson. Okay, so people or like them do not have any idea how the DNA replicates. So they develop, alright, so these biologists develop several models on how the DNA replicates. Alright, so okay, develops a model on how the DNA replicates. We have right here 3 models. Alright, and these models that we have right here are the conservative, semi-conservative, and the dispersive models. Okay, now what is the difference between these 3 models that we have? And in reality, what kind of model does the replication process in our cell follows? Okay, so let's start first with the conservative. Alright, so the conservative one, from the word itself, conservative means to conserve. Okay, it conserves something. Number 1, the process, and the next one is the DNA itself. Okay, so in this case, after the first replication, the parent DNA was retained. Okay, the parent DNA was retained, and it produces a newly synthesized DNA over here. Okay, basically speaking, in the conservative manner of replication, nothing splits. Okay, nothing splits. Only the DNA of the parent, or the parent DNA, will produce its own and the new one. All right, and so since it produces its own, all right, its own copy, so this is what this is what we meant by conservative. So it produces its own copy. So it doesn't be deleted or something like it is not something like erased. All right, so something like that. Okay, now when this particular DNA molecule, just like its own parent, all right, just like the same thing as the parent, when this one replicates for the second time, it will do the same thing the parent does. Okay, it will divide or it will replicate to produce its own, the same thing, and another newly synthesized DNA. All right, but the newly synthesized DNA, the one that is right here, so when it replicates, obviously and logically speaking, it will produce its own. All right, it will produce its own. It will not produce any copy from its parent. Only it will produce its own kind. Okay, as you can see here, it will produce only red. Okay, so that is what we meant by conservative. Okay, so number one, the molecule was conserved and the process is also conserved in the long run. Okay, so that was the conservative method. So the next method is the next model is semi-conservative. So what do we have here in the semi-conservative? So do not look at the red ones here. So do not look at the red ones. So we have here the parent DNA molecule. Alright, so we have the parent DNA molecule and these two will split. Alright, so one over here and another one over there. Now, these two parent DNA molecule will create, right? So it will create a newly synthesized complementary pair. So this is its complementary pair. Okay, so the second strand will also create a complementary pair, which is a newly synthesized strand. All right, so the one that we have right here. So this one and the other one on the other side. All right, this time, why is it called semi-conservative? Okay, why is it called semi-conservative? Because in this model, the original DNA, which is coming from the parent DNA over here, will be preserved or will be conserved. Even though it undergoes several replication processes, the parent DNA molecule will be continued to be conserved. Okay, in the process. Okay, so for example, again, let's go to the second replication. If this one splits, all right, if these two splits, one over here and one over on the other side, so again, it will follow the same process. Okay, so the number one will follow the same process, the original process on the first replication. It will produce a newly synthesized strand over here. Okay, so the newly synthesized strand over here, and the number two obviously will produce what kind of strand? The same thing. If it is red, it will produce the same thing. Okay, so on the other side, it's doing the same thing. So the parent DNA molecule, the parent DNA molecule will follow, all right, so will follow the same process. It will produce, or it will split up, one here and another one over on the other side, and it will follow the same process. It will synthesize a newly it will synthesize a new strand over here. Okay, it will synthesize a new strand, and the red ones obviously will produce a red one. All right, so that's what we meant by semi-conservative method or model. Okay, that we have right here. Now, meanwhile, in the dispersive model, right? In the dispersive model, nothing splits up. Okay, during replication, unlike in the semi-conservative, nothing splits up. Okay, instead, from the word itself, dispersive, the parent molecule, all right, so the parent DNA molecule is dispersed or randomly distributed on the daughter DNA molecules. Okay, the one that you like the one that we see here. Okay, so the parent molecules or the DNA molecule coming from the parent was distributed randomly on the next generation. Okay, so assume that this undergoes mitosis. Okay, so probably the parent molecule will be distributed randomly. Now, as the generation progresses, okay, as the generation continues, what happens to the parent molecule is it begins to shrink, okay, in size as the newly synthesized segment of the DNA is continually to be synthesized in the process as the generation continues. Okay, so as you can see here, again, the parent molecule has been randomly distributed all throughout the generation, but it doesn't look like on the second generation. It is now being cut short. Okay, it is shortened up, okay, compared to the previous one. Okay, so that is the dispersive model. Now, what do you think? Okay, what model do you think our DNA replication or our cells in the during the DNA replication follows? Okay, so what kind of model are we? What kind of model are the cells following? Okay, so technically speaking, it follows the dispersive the semi-conservative method. Alright, it follows the semi-conservative method, and just like what you see on the diagram that we have right here, so definitely it is a semi-conservative method. Why? It's because what you have right here, so from one DNA molecule, from one parent molecule, it becomes now two daughter DNA molecules. And as you can see here, from the parent molecule, the violet one over there, it produces a newly synthesized strand over here, the blue one. Alright, so the blue one that you have right here is the newly synthesized strand of the DNA. Okay, so in order for this process to work, okay, there are a lot of things that is needed. Okay, so which I will show you later. Okay, and what is the most important thing that we have right here is the replication process. Okay, the replication process itself. So we have here the replication fork. The direction is always opposite to each other, one from the left and one from the right side. Okay, then eventually these two daughter strands that you have right here, these two daughter strands will connect halfway. Okay, so they will connect halfway, and it forms now your two DNA molecules that you have right here, the two daughter DNA molecules. Okay, so this is how the replication works, and it follows the semi-conservative process. Alright, so what are the things needed in the DNA replication? The first one is the helicase. So as we all know, the helicase is the one that remodels the DNA. Now in this case, the helicase is just like a zipper. Okay, as we all know that the DNA is a double-stranded molecule. DNA is a double-stranded molecule, and okay, what it does in order for this double strand to be unzipped is a helicase. Okay, the helicase separates the two strands of the DNA that is connected by the hydrogen bond. They are connected by hydrogen bond, and the helicase is the one that separates it. Okay, just like a zipper. When you pull down your zipper, the two parts of the zipper separates. Okay, now after the separation, here comes now the DNA polymerase. The DNA polymerase is now currently synthesizing. It is now currently synthesizing the strand, okay, in order to return the DNA into its double helix format. Okay, so as you can see here, it collects and it consolidates the free nucleotides in the nucleus. And once it is done, you now have a double-stranded DNA, and not just one but two double-stranded DNA as part or after the that is the product of the replication process. Okay, so again, what we need right here is a helicase that will remodel the DNA, and we have the DNA polymerase which synthesize the new strands that we have right here. Alright, so that is the materials needed for DNA replication. And what does it look like in the chromosome? So as we all know, chromosome is made up of DNA, right? So the chromosome is made up of DNA. So originally, the chromosome looks like this before cell division. Okay, it has one chromatid per chromosome. So all chromosomes in our body has one chromatid. But after replication, that one chromatid becomes now two chromatids. Alright, so we called it the sister chromatids. Okay, so we have now the two chromatids. Then after mitosis or cell division, these chromatids will separate. Alright, so these chromatids will separate in the anaphase. Alright, particularly in that particular phase of cell division, it will separate on the anaphase and it will go back again to its original form. Okay, it will go back again to its original form. So this is what it looks like in the chromosome. Okay, so now let's now discuss what is the goal of the DNA replication. All right, the goal of the DNA replication is very simple. Okay, to make all somatic cells carry the same genetic information. That's all. Right? So what are these somatic cells? These are body cells. Right? These are body cells. When we talk about body cells, it doesn't include the egg and sperm cell because they are gametes. Okay. Now what are these body cells? Skin cells, bone cell, brain cell or nerve cells that we have, muscles, okay? Smooth cells in the smooth muscles, something like that. Okay, cells in your digestive system, red blood cells, white blood cells, so on and so forth. Except egg and sperm cell. Okay. Now, all of those cells that I mentioned must carry the same genetic information, whatever genetic information do you have on your body. Okay? So, in short, the goal of the DNA replication is for first one, growth, and the second one is for maintenance. Alright? So, what is important here is the maintenance of body parts. So, logically speaking, if you will going to repair the body parts that was destroyed or broken, if you will going to repair those body parts, the body parts that you will replace should be the same thing as what it is destroyed or broken. Okay? So, I'll give you one example. Okay, I'll give you one example. Example, the skin. If the skin or if the replication fails to carry out the same genetic information, the skin color that you have right there will be completely different from the surrounding skin that you have. Okay, for example, you are dark colored skin and suddenly the replication process fails to do that. Okay, the replication process fails. So what happens is there are skin cells, or there will be a skin cell that are color white on your body. So it will look ridiculous, right? So those people who has vitiligo, something like that, that is a disease in which the coloration of the skin cells is different from the surrounding skin cell. All right, so that is an example of what will going to happen if the DNA replication fails to do its job. Okay? It fails to do its job, especially during the maintenance process. Okay? So from the tip of your hair to the tip of your toes, the DNA components, okay, the components of the DNA of each cells in your body must be the same all throughout. Alright? Except for the egg cell and sperm cell. That is a different forms or classification of cells. Alright? So this is the importance of why we need DNA replication. Alright? So up next is the main topic that we have right here, which is how proteins were made. Okay? So let's try to discover that one. Alright, so here we are in the part where we will going to discuss or discover how the proteins were made. Okay, so we called it the protein synthesis. Okay, that's the exact word that we have right here. Now, what are these proteins? Okay, so basic trivia about the proteins. Proteins are made up of amino acid. That is their building blocks. Okay, so the proteins building blocks are called amino acid, and these amino acids are connected via peptide bonds. right? So if you see a connection between one or more amino acids, that is the peptide bond that do that particular connection. All right. So as the amino acids get longer and longer, they are now called polypeptides. All right. They are now called polypeptides, and these polypeptides eventually will form a protein. Now, where can we find protein in our body? So, the proteins are found in your eyes, the eye color, all right, the skin color that you have, all right. So, the characteristics that you have on your body, like shape of the nose, okay, shape of your ears, okay, the body feature that you have, yan. So, all of those, especially the muscles, alright? Muscles, and you have the fingernails and your hair as well, is made up of protein. So, that is the protein, including hormones, okay? I forgot to mention, the hormones and the enzymes that are found in your body is also a protein. That is how important these molecules that we have right here. Proteins are part of the 4 biomolecules. Okay, proteins are part of the 4 biomolecules, and proteins were made during protein synthesis. Okay, so that's the function of the protein. Okay, in our body. So the question here is how it is made. What are the things needed, or what are the, aside from the things needed, what are the process needed in order to create one of these proteins? Okay, so let's discover this one. Alright, so before we go to the main process, let me prime you with some ideas that we have right here. So we have here the DNA genotype is expressed as proteins, which provides the molecular basis for the phenotypic traits. So as we recall, okay, what do you mean by the genotype? Okay, and what do you mean by the phenotype? Okay, so the genotype is the genetic or the molecular form of the trait. Okay, example. Okay, if you recall your grade 8 lesson about the Punnett square and Mendelian genetics, so if you will recall that the capital letter represents for the dominant trait, and the small letter represents for the recessive trait. For example, we will use the height, okay? The height of the plant, okay? Yeah, right. So the height of the plant. So we have it tall, and we have it short. All right. So this is the height of the plant. All right. So what do you mean by genotype, and what do you mean by phenotype? All right. So if we write this one, if we write this particular trait, like the one that we have right here, okay? If we write that one like this, we will call this trait as heterozygous tall. Alright, so we will call this trait as heterozygous tall, or simply tall. Now, what is the genotype here? The genotype is this letters that we have right here. This is the genotype. And the phenotype is the physical appearance. Now, you cannot see the genotype. Alright, you cannot see the genotype because the genotype is located on the DNA molecule. Alright, that is part of your DNA molecule, the genotype, together with the other traits found in your body. So that's the genotype. Now, what is the physical manifestation of this genotype? We called it the phenotype. Alright, so the phenotype is what the other sees. Alright, what you see on that particular organism. For example, you see the eye color of that person that is blue. So that is the phenotype. But you cannot see what kind of, you cannot see the genotype of that blue eyes. Only the physical manifestation. Alright, so the blue eyes, that's the physical manifestation. Alright, so that's the difference between genotype and phenotype. Now, by the way, I forgot, we call these traits that you have right here, we called it alleles. Alright, alleles are forms of the genes. Okay, so there are two forms of your genes. We have the dominant and the recessive ones. Okay, so not to mention also some incomplete dominance and codominance, alright, in the process. Alright, so there are two main stages of creating proteins. The first one, it's the transcription, alright, And the second one is the translation process. Okay, we will go into discover that one later in this topic. And the next one, genetic information is written in codons. So if you forgot what is codons, it's what is codon is all about. Codons are basically the segments of the DNA. Okay, codons are segments of the DNA that is composed of 3 letters. Alright, so this codons is composed of 3 letters, and these 3 letters corresponds to the amino acid sequence that they will produce. Okay, so basically that's how it works. For example, I have right here U, A, G. Alright, so codons is always made up of 3 DNA bases. Alright, or bases, something like that, not DNA bases. It is made up always of 3 bases. So 1, 2, 3. And these 3 bases corresponds to a or an amino acid. Alright, so to give you an idea, so I have if I have 10 codons, how many bases do I have? Alright, so there are 30 bases. Alright, so that's how it works. Okay, so if I have 5 codons, how many amino acids do I have? Okay, how many acids will I be able to create? So I have 5 amino acids. Alright, so that's how it works. Okay, so codons. Codons are very important in the topic that we have right now. Okay, so as we all know that the adenine is paired with thymine and the cytosine is paired with guanine in the DNA. But what about this molecule? Alright, so what about this nitrogenous base, the uracil? Where can we find them? Alright, so you can write your answer in the comment section below this video. Alright, so we can find the uracil in the RNA. Alright, so we can find the uracil in the RNA, and usually the RNA is or the uracil is paired with adenine. Okay, in short, the uracil is the counterpart or the opposite of the thymine in your RNA. Alright, so it looks like this. Okay, in the RNA. So adenine will be paired up with uracil, alright, in RNA, and the thymine is only found in DNA. That's their difference. Okay, so now what we have right here is the genetic code chart. Okay, so what is the genetic code chart? This is the collection of amino acid and codon bases that corresponds to your amino acids. Okay, so there are 20 of them. Okay, there are amino acids that will trigger. Okay, there are amino acids that will stop the process of protein synthesis, and there are 20 of them all in all. Alright, so those 20 amino acids are being produced using certain codons, as you can see here. The codons that you can see here represents the amino acid that it belongs, or it is translated. Okay, so in this codon chart that you have the first base, you have the second base, and you have the third base over here. So how does it work? Okay, so let me teach you how this codon chart works. Okay, so for example, you have a codon of RNA. So RNA, we follow the RNA here, not the DNA. Because in DNA, there's nothing, there's no base there that is written as U, okay, on the DNA. Alright, so suppose that you have codon AGU. Alright, suppose that you have a codon AGU. So the question here is what amino acid it is translated. What kind of amino acid it will be translated. So if you take a look at the first base, we have here letter A, the second base will be letter G, and the third base will be letter U. Okay, it will be read as from left to right. Okay, so from left to right, it is the way of reading the codons that you have right here. It is from left to right. So let's start first with what kind of amino acid it will form. Okay, let's start first with the first base that you have on this example. Okay, so the first base is A. Alright, so the first base is A, or adenine. So, meaning to say, the first base will determine what row are you going to locate your amino acid. Alright? So again, the first base will determine what row, alright, are you going to locate your amino acid. Next one, the second base was G. Alright, so G. Now, the second base determines what column. Alright? The second base determines what column your amino acid located. Alright? So, in this column, okay, that you have over here. Next one, the third base is the more detailed. Alright? It will give you the specific amino acid, the specific location of the amino acid. So, we have here the first base, second base, and the third base is letter U. Alright, so the third base is letter U. So, in short, the AGU, alright, the AGU is the codon for, alright, the AGU is the codon for serine. Okay, so the AGU is the codon for serine. Alright, so let's have another example. Okay, suppose you have a codon called GUU. Alright, suppose this is your codon. Now, the question is, what amino acid does this codon translated? Alright, so what amino acid it is being translated? Okay, so let's take a look at this codon. So first, let's have this first base. Okay, so that was G. Okay, next one, we have the U, the second base, so this column. And to be more specific, what amino acid are we looking for? So we have the third base, we have the U over here, and that's the GUU. Okay, so the GUU is corresponding to the amino acid called valine. Alright, it corresponds to the amino acid called valine. Okay, so that's the GUU. Okay, so that's how you use the codon chart that we have right here. Alright, so we have here the Rosetta Stone. Okay, now in order for you to understand the Rosetta Stone, by the way, Rosetta Stone is an artifact from ancient Egypt. It contains some it contains some rules and laws, okay, during those ancient times. Okay, and this written language that you have right here. So to tell you honestly, I don't understand anything regarding to what I see on this picture that we have right here. But there is a person who can interpret this one. Okay, so an expert Egyptologist who can interpret a language written on this Rosetta Stone. Okay, now once that person who is an experienced Egyptologist and be able to interpret what is written in the Rosetta Stone that you have over here, it will now be translated to a more understandable language, okay, that we have right now. So probably that is the English language. Okay, right? So it will be translated on that, and we will be able to understand what is written on this Rosetta Stone tablet. Okay, now that's the same thing. That is the same thing the cell follows. Okay, there is someone who will interpret, and that interpretation will be translated to a more language that is common all throughout inside the cell. Okay, so how it does? Okay, what process does it follow? So we have here the first one called transcription. All right, it is very simple. The transcription process. All right, so the transcription process is the process that creates. Okay, so from the verb right here, creating. Okay, so it creates the equivalent RNA copy of the sequence of the DNA. As we all know, DNA is located in the nucleus. Alright? So, why do we need to get the copy of the DNA? Because the DNA doesn't, it is not designed to get out of the nucleus. Alright? It is not designed to get out of the nucleus. So, that is why you have your, you have a certain reagent that is needed in order to create the copy of the RNA or to create the copy of the DNA, which is located on the nucleus. Okay? And eventually, after the process of transcription, what is the output? The mRNA, which is needed on the next process. Okay? So the mRNA is very important on the next process. Now, technically speaking, what template do we need here in order for the transcription process to work? What template do we need here? The DNA. Okay? If you don't have the DNA, then the transcription process is useless. Okay? What do you need, or what will you transcribe if you don't have the template to be transcribed with? Alright? So that's the logic here. Okay? So the DNA is very important in creating the mRNA. Alright, so let's take a look. Okay, so this is the transcription process in the nutshell. Alright, in a nutshell. So, as you can see here, the DNA is being copied by the RNA over here, and eventually, it will produce the mRNA that we need for the next process. Okay? Now, this is the picture on how the transcription process happens. Okay, so to further understand on how the transcription process works, okay, so I will give you first the things that you need to understand. So what is a gene? All right, so what is a gene? Gene is a segment of DNA. All right, so gene is a segment of DNA. A segment of DNA. Now, depending on what kind of gene is that, so it can be long or short. Alright, so that's the length of your gene. Alright, and the genes are made up of codons. Okay, several codons. Okay, so depending on its length. And the codons are made up of bases. So therefore, the genes are made up of several bases of your DNA. Now, the genes are very important in this process. This is the main template. Alright, this is the main template that the RNA polymerase will going to use in order to create the mRNA. Alright, so to further understand how this one works, we will going to use a simulator, courtesy of PET simulation. Alright, so PET simulation generously lets us use this kind of simulation that we have right here. So you can go to their website. It is written in the description below this video. You can visit the website and download this simulation there. And also, you might want to donate, okay, for their cause in order to create more of this simulation in the future that will help students, okay, that will help students all over the world in understanding science and mathematics. Alright, so now let's discuss how the transcription process works. Okay, now the things needed for the transcription process are the following. The first one is the transcription factor. Alright, so logically speaking, the transcription factor is for transcription. Alright, so the transcription factor is a factor or is an enzyme that triggers or suppress the gene expression. So by the way, why do we need to express the genes? The reason is to create proteins. Okay? The reason why we express the genes is to create proteins. Number one is the transcription factor. So transcription factor is very important. Okay? It can trigger, it can suppress the gene expression. Alright, for example, I have right here a positive transcription factor. Alright? And what it does is it triggers, right? It triggers the gene expression. Okay, it triggers the gene expression that we have over here. Okay, so the other one that we have over here is the negative transcription factor. So the negative transcription factor, it suppresses the gene expression. Okay, so another important thing that is needed in the transcription process is the RNA polymerase. Okay, the RNA polymerase is the one that synthesizes. Okay, it is the one that synthesizes the mRNA later on. All right, so let's take a look if we will going to create an mRNA using the transcription factor that we have over here. Okay, so let's put it that one more time. Okay, so as you can see that the negative transcription factor is the one that suppress the gene expression. Okay, so let's put now the positive transcription factor. Let's put the polymerase over here, and as you can see, it elongates. Okay, so as you can see, it elongates and it creates now your mRNA. So this is the mRNA. So it goes out of the nucleus. Alright, it goes out of the nucleus and proceeds to the next process. Okay, so let's take a look at the other genes. Okay, so this gene is much longer. Alright, it is much longer than the other one. Okay, so the question here is, does the length of the mRNA depends on the length of the gene? Okay, so I repeat the question. Is the length of the mRNA dependent to the length of the gene? Alright, so you can write your answer in the comment section below this video. So definitely the answer there is yes. Right, the answer there is yes. Why? It's because the longer the gene that you have right here, the longer the RNA polymerase will going to synthesize that particular mRNA. Okay, so in this case, we have a longer gene over here. So it requires 2 positive transcription factor. Okay, so if you're going to use one transcription factor, if you were going to use one, it will not express the gene. Okay, so it will not express the gene. Otherwise, by putting another transcription factor, that's the time your RNA polymerase will do its work. Okay, it triggers. It requires 2 transcription factor to trigger the transcription process. Okay, so there you go. Okay, so if we put that one more time here, it will now create another mRNA. Okay, so there you have it. So let's try to look at the other gene. Okay, so in this case, it requires another transcription factor. It requires 2 transcription factor, and as you can see, it is much longer than the other one. Alright, it is much longer than the previous one. So if we put some RNA polymerase over here, okay, if we put some polymerase and it will do its function, so it synthesizes a much longer mRNA, and the length of the mRNA is based on what we have on this gene over here. Okay, so that is the transcription process. Okay, so that is the transcription process over here. So okay, it looks like this in a more summarized. Okay, in a summarized way, it looks like this one. Okay, so the red one that you have right here, that is the mRNA that was formed on the RNA polymerase, and it uses the DNA as a template. Okay, so that is the transcription process. Alright, so we have here the next process called the translation process. Okay, so after interpreting the DNA template that you have in the first process, so we are now going to translate them into a more functional molecule called protein, or the amino acid first before it becomes a protein. Okay, so just like in real life, if you will going to read a book, you need to interpret what is the book is trying to say, what is the particular paragraph and statements inside the book is trying to say, and you will going to translate them in your own words. Okay, for you to better understand it. It's the same thing that is happening inside the cell. Okay, so aside from interpreting what is on the DNA template, it should be also be translated into something more useful. Alright, so this is the first stage of protein synthesis. Okay, this is the first stage, not the transcription process. Okay, so the translation process is the first stage of protein synthesis, and in this process, the production of the proteins is being done. Okay, the production of the protein is being done. Now, with the use of the template that comes from the DNA, which is in a form now of mRNA. Okay, so in a form now of mRNA, So with the use of the mRNA, we will now be able to produce the amino acid needed to create these proteins for the cells that we have. Okay? So now, where's the translation process occur? Now, it occurs in the cytoplasm because this is where the ribosomes are located. Okay, this is where the ribosomes are located. Now, together with the ribosomes, there are things that are also needed, which is called the tRNA. Okay, so the tRNA, it is also needed in this process. So later, I will show you how this ribosome and tRNA works in order to create this functional product called protein. Alright, so okay, so for you to better understand how the translation works, okay, let's go back to our simulation again. Okay, again, we go back now to our simulation right here. So let's go reset once more this simulation, and the things that we need for the translation process is in here. We have the ribosome, and the mRNA destroyer is is somehow needed in the process. Okay, so I will show you later how this one works. Okay, so the first thing that we have right here is the transcription process. So in the transcription process, you need a transcription factor, an RNA polymerase, then it creates the mRNA. Okay, so if you do the process several times, you will produce several strands of mRNA. Okay, there you go. Okay, so we have now a lot of mRNA, so we need to translate them to become a functional protein. So using the mRNA strand that you have, we will now be able to translate them into a protein that I'm holding right now. Okay, so the protein, right? So if we put some more ribosomes in the system, so we can now create a lot of proteins. Okay, but there's a drawback here. Okay, there's a drawback. This is an energy. This particular process consumes a lot of energy. Okay, this particular process, the protein synthesis, it consumes a lot of energy. So the thing here is, the body only creates protein when it is needed. Okay, the cells will only create a protein when it is needed. It is the proteins are not always created 24/7. Okay, so what it does is the cells contain some mRNA destroyers, and it destroys the mRNA in order for the cell not to create more proteins. Okay, so the cells doesn't need a lot of protein. It only needs it only creates when it is needed. So the cells have has a protocol to destroy all of this mRNA that you have on your cytoplasm in order for not to create a lot of proteins here. Okay, so the next gene that we have over here is a longer version of the previous one. So again, you need a 2 transcription factor. Okay, so again, if we put some negative factors here, if we put some negative transcription factor, it will not do the transcription process. So again, with the use of the polymerase that you have right here, it will create the mRNA necessary, or it will create an mRNA for this gene that you have over here. Okay, now the question here is this the same protein that we have on the previous gene? Okay, so let's take a look. Okay, so the protein that we produced over here is completely different from the previous one. So it tells us that the third gene is completely different from the second gene. So the protein from this third gene that you have over here is different from the previous one. Okay, because in terms of length, it is somehow longer. It is somehow longer than the previous. Okay, so Okay, so there you go. So this is a complete, this is a different type of protein that you have right here. Okay, so in short, we can say that the genes, okay, that the segment of the gene or the segment of the DNA tells what kind of protein are we going to produce. All right, and it shows us that not all genes are equal. Not all genes are the same. They are different entirely all throughout in our body. All right, so it might be that this genes is for eye color, it might be that this genes is for height, and so on and then so forth. All right, so this is how the translation process works. Now if you're going to ask me, sir, where can we find the tRNA? The tRNA is inside this ribosome. All right, the author of this simulation did not bother to remove the tRNA from the ribosome. It is in there. Okay, so it is in there. Now, to further understand what is more all about the translation process. Okay, so the tRNA is only the, okay, from the word itself, transfer RNA. So it only transfers the amino acid. It only carries the amino acid. That's all. It doesn't do anything aside from carrying the amino acid. Now, the codons the tRNA contains is called the anticodon. All right? So why is it anticodon? Because it is anti to the mRNA. The codons that are carried by the tRNA are opposite to the mRNA. All right? Now, if you're going to take a look at this one, all right, if you're going to take a look at this one, the amino acid that you have right here follows the template from the mRNA. Okay, I repeat, the phenylalanine that you have over here follows the template that was written on the mRNA, not on the tRNA. Okay? Now, if you want to find out, alright, so if you want to find out if UUU is the codon for lysine, let me know in the comment section below. Okay? So, try to find out if UUU is the codon for lysine. So, try to find it out using the codon chart. Okay? So, again, the mRNA is the one that is used to follow the amino acid that will be created. Okay? So, we have right here the more the step-by-step process. Okay? So it starts first with the anticodon approaching the mRNA over here in the ribosome, and as it does, it creates the chain of amino acid, and now to be called polypeptide chain in the process. Okay, and once it encounters a stop codon, literally it will stop. Okay, it will stop doing the process. It will stop creating the amino acid. Then therefore, the length of the amino acid or the length of the protein is dependent on the length of your mRNA that was transcribed. Okay? Or that was translated. Okay. Alright, so to summarize the process between transcription and translation, I made a Venn diagram for you to understand their differences and similarities. So the differences that you have right here is the transcription is the product of the transcription is mRNA, whereas in the translation the product is protein. All right, and in the transcription it occurs in the nucleus. Meanwhile, in the translation process it occurs in the ribosomes. Okay, which is if we go to the transcription process, it requires the RNA polymerase for it to work. Meanwhile, in the translation process, it requires a lot of reagents like ribosomes and tRNA. Okay? It requires ribosomes and tRNA in order for the translation process to work. Okay? Now, what are the similarities between these two processes? Okay? Although they have differences, they also have similarities. Okay? So, both of them uses nucleotides. Alright, both of the process uses nucleotides. Cytosine, guanine, adenine, uracil as their nucleotides. Okay? So, and most important, the similarities between the two, they need a template. Okay? Without the template, transcription process and translation process will not work. Now, what particular template? So, this one, the template here is DNA. And on the translation process, the template is the mRNA. Okay, so that is their differences. Okay, so that is the two processes that we have right here. And furthermore, if we were going to summarize the whole process that we have encountered today about the protein synthesis, it starts first with the transcription process on the nucleus. And as I've said earlier, it requires a lot of energy. Alright, it requires a lot of energy to attach the amino acid to the tRNA that you have over here. Okay, so it requires a lot of energy, and after the mRNA has been translated, it must be destroyed immediately. Okay, it must be destroyed immediately for it not to cause the cell to build up more amino acid on its system. Okay. And the tRNA is the one that carries out this process. Okay, it carries out this process, and also it carries out the amino acid that the transcription or the translation process produced in the end. Okay, that you have over here. So it has the start codon, and then it has the stop codon that stops the overall translation process, and that's how you create proteins. Okay, in this with the use of these two processes over here. Okay, so that was the transcription and translation process of protein synthesis. All right, so for me to know if you understand what the concept I have discussed about the protein synthesis and the process behind that one and how it works, so let me know in this practice problem if you did understand all these concepts. Okay, so I have right here a certain DNA strand. Okay, and in this practice problem, you need to find out the complementary DNA strand of this one. Okay, the one that you have over here. And you need to find out the mRNA. And you need to find out the tRNA. And also the amino acid of that DNA that you have, the DNA segment that you have over here. Alright, so let's try. Let's try to answer this one. So if you want to have more time thinking and answering this particular practice problem, you can pause this video, all right, if you want. Okay, so let's start first with the DNA that you have over here. So if this is 5 prime, so we will going to have on this side 3 prime. Okay, so what is the complementary pair of the A on DNA? We have T. We have A over here. The G will be complementary to C. Okay, next one is C. We have A. We have G. We have C, C, and finally we have G over here. And if this is 3 prime, then therefore the last part will be 5 prime. Okay, so later I will discuss that one. What do we mean by 3 prime and the 5 prime over there? Okay, so let's put this one into a bracket to make it more simplified, to make it to show that it is in a form of codon. Okay, so let's take a look at the mRNA. Okay, what is the mRNA sequence for this segment that you have over here? What is the mRNA sequence for this segment? Okay, so if this is 5 prime, so the mRNA should be in 3 prime. Okay, so we have A, okay, so A is U on the RNA. So we have A, C, then we have C, A, G. Okay, then followed by C, C, G. And that is also 5 prime. Okay, so let's put it into a bracket to show that they are in a form of codons. Okay. Alright, so that was the mRNA. So did you get it right? Okay. So next one is the tRNA. So the anticodon. So again, these are the anticodon of mRNA. Okay? So the anticodon of mRNA. So let's put it this way. So the U is A. So as you can see, we will not use the 3' and the 5' here, ha. Okay? So it is not needed anymore on the tRNA. Alright, so we have U, we have G, we have G, we have U, and we have C. So we have G, G, C, and bracket for the codon, and then that's it. Okay, now the challenging part here is what is the amino acid? Okay, what is the amino acid translation of this segment? Okay, so again, the process will be using the mRNA. We'll be using the mRNA to find out what is the amino acid. Okay, so we will be using mRNA to find out what is the amino acid. So let's take a look at this amino acid using the codon chart. Alright, so we have here the UAC. So let's take a look on this codon chart. Right, so let's take a look at this codon chart. Where is the UAC here? UAC. Alright, so that is tyrosine. Right, so that is tyrosine. Alright, so next one, we have the CAG. Alright, so we have the CAG. Okay, CAG. Alright, so that is glutamine. So that is GLN. Alright, so that was GLN. Alright, so next one, next group is the CCG. Alright, so next group is the CCG. Okay, CCG. Alright, so that is proline. Alright, so proline. Alright, so did you get the same amino acid that like the one that I have right here? So if so, just comment your answer. Alright, so in the comment section below this video. Alright, now what do we mean by 3' and 5'? Alright, before we go to the next set of practice problem, what do we mean by 5' and 3'? So these primes that you have right here represents the number of carbon. Alright, it represents the number of carbon in which the phosphate group and the hydroxyl group is being attached. Again, this is the number of carbon. Alright, the number of carbon in which the phosphate group and the hydroxyl group of your DNA is attached. For example, why is it called 5'? Because the phosphate group is attached to the 5 or the 5th carbon in this DNA molecule. Alright? The 5th carbon in this nucleotide. Okay? So, now, the counting is always clockwise. Okay? Starts with 1, 2, 3, 4, and 5. So, that is 5'. Next one, why is it called 3'? Okay, why is it called 3'? Because the hydroxyl group is attached to the 3rd carbon of your nucleotide. Okay? Again, the hydroxyl group is attached to the 3rd carbon of your nucleotide. So if we're going to count this one, 1, 2, 3. So that's the 3rd carbon. Okay? So therefore, that is 3'. Now, whenever you have 5', on the other side should be 3'. Now, if you have on the segment, if you have 3', the complementary side should be, okay, the complementary side should be 5'. Alright? So, that's how it works. Okay? So, that is what the 5' and 3' stands for. Okay? Now, let's go to another practice, set of practice problem. Okay, so I have right here this problem. Again, you can pause this video if you want to have time thinking and answering this practice problem that we have right here. Okay, so let's find out first the complementary strand of the DNA. Okay, so again, if this is 3', the complementary strand should be 5'. And if this is 5', the other side should be 3'. Okay, so let's put the complementary pairing that we have over here. So we have T, we have G, we have C, we have A, C, then we have T, we have a G over here, and we have G over here. Okay, so let's check this one out one more time if we are correct. So bracket, bracket, bracket for codons. Okay, next one, we have the mRNA. Okay, so the mRNA, so we have U, G, C, we have A, C, and we have U, G, G. Again, 3 prime and 5 prime. Okay, so did you get the right answer, guys? Alright, so did you get the right answer? So write it down in the comment section. Okay, if you don't have any problem about getting the correct answer for this problem. Okay, again, codons for, or brackets for codons. Okay, again, let's take a look at the tRNA, the anticodon of mRNA. Okay, so we have A, C, G. Okay, we have G. Alright. Right, so let me erase this one. So UUG. Okay, so next one we have A, C, C. Okay, so brackets for codon. Okay, so we are done with the tRNA, and let's take a look at the amino acid that is translated, okay, on that DNA segment. Okay, so the first one was UGC. Okay, so let's take a look. Alright, so UGC. U, G, C. Okay, so that was cysteine. Okay, so cysteine. CYS. Alright, so next one is the AAC. Okay, so let's go back to the codon chart. AAC. C. So that was asparagine or asparagine, ASN. Alright, so we write here the abbreviation ASN. And finally, we have the UGG. Okay, so what does UGG amino acid translated? Okay, UGG. Alright, so that was tryptophan. Alright, so TRP. Alright, so that was tryptophan, TRP. Okay, so there we have it, the amino acid on our problem set that you have over here. So did you get it right? Okay, so well, good for you if you got it right, and if not, try to practice more. All right, try to watch again the concept if you missed out a concept and you are confused right now. Okay, so don't worry, we have another practice set that we have over here. Okay. So, in this case, we have a new segment over here. We will do the first process, find the complementary strand of the DNA. Okay, so we have here 3 prime. So, in short, this will be 5 prime and this will be 3 prime. Okay, so we have here T, A. So, we have G, A, C, and we have C, G. What do we have right here? A. So we have T. Alright, so there you go. Okay, so 3 prime and 5 prime. So brackets for codons. Okay, so how about the mRNA? Okay, so the mRNA. So we have U, A. So we have G, A, C. And we have C, G, U. Okay, again, 5 prime and 3 prime. Okay, so codon, brackets for codon. Again, tRNA. tRNA is an anticodon of mRNA. So let's write this one up. So U. Okay, so we have CUG and we have GCA. Alright, so codons, brackets for codon. Okay, so what you're going to do here is you need to practice, okay? You need to practice doing this one over and over again for you to be familiar with the process. Okay, so again, let's use the genetic code chart for our amino acid. For those who are still confused, mRNA is the one that is used to make the amino acid. Okay? We will use the mRNA template to create this amino acid or to be translated as amino acid. Alright, so let's take a look. That was UUA. UUA. Alright, so UUA. Alright, so that was leucine. Okay, so LEU. Alright, next one, we have GAC. Okay, so we have GAC. So, going back to this codon chart, GAC. Okay, so that was aspartic acid. So, ASP. Alright, so we have ASP. Next one, we have CGU. Over here, CGU. CGU. Alright, so CGU. Alright, so we have arginine. Alright, so we have arginine. Okay, so these are the set of the amino acid. Okay, so did you get it correctly, guys? Okay, so we have now the last part or the last set of practice problem. Okay, so I hope on this fourth practice problem you are comfortable now on solving this particular problems about protein synthesis and DNA replication. Alright, so without further ado, we will not waste time anymore. So let's try to find out the complementary strand of the DNA that we have over here. Okay, so this is 5. So this will become 3 and this will become 5. Okay, so we have C, A, C. Alright, so on the other side, we have G, A, G. And also, we have C, E, G. Alright, so brackets for codon. Okay, next one, we have the mRNA. Okay, so the mRNA, so again, if that is 5 prime, so the mRNA that you will have is also 3 prime. Okay, so 3 prime and this one is 5 prime. Okay, so we have C, A, C. We have G, A, G. And also, we have C, U, G. Okay, so bracket. Okay, next one is the tRNA. Okay, so the tRNA, so we have G, U, G. So we have C, U, C. And we have G, A, or okay, we have G, A, C. Okay, so codons or brackets for codon that you have over here. Okay, so lastly, we will going to use the mRNA to find out what is the amino acid using the codon chart. Okay, in this case, we have the CAC. Okay, C, A, C. Okay, so that was histidine. Alright, so CAC stands for histidine. Okay, next one, we have the GAG. Alright, so this is the GAG. Right, so GAG. GAG. Alright, so we have the glutamic acid. So we have the glutamic, so GAG, GLU. Stands GLU for abbreviation, glutamic acid. And finally, we have CUG. Okay, so we have the CUG over here. Okay, CUG. Alright, so there you go. So CUG. So that is CUG. Alright, so that was leucine. Okay, so that was leucine. CUG stands for leucine. Okay, so there you have it. Okay, so did you get all the 4 practice problem set that we have right here? Did you get all of them? Okay, so if so, then congratulations to you guys. You'll be able to understand the concepts behind this lesson that we have on this video lesson. Alright, now to summarize everything that we have in this process. Okay, so we call this process, the overall process that we discussed today, we called it the replication and the protein synthesis. We called it the central dogma. So what do you mean by the central dogma? Central dogma was first identified by Francis Crick, and he described the central dogma as the process, okay, inside the cell that is used by the cell in order to create something that is functional, from DNA to something that is functional. What is this something that is functional? And that is protein. Okay, so that is protein. This is the functional molecule. that is described on that particular on the definition of the central dogma. Again, central dogma is the process in which the DNA is converted into something functional, a functional molecule. In this case, we have protein. Alright, so we have here the replication. Okay, so the DNA will replicate itself to become DNA. DNA. Next one, RNA also replicates. Okay, in a special conditions, RNA also replicates. But the overall process, the transcription and translation is in here. So all cells, bacterial cells, protozoan cells, or something like eukaryotic or prokaryotic cells, it follows the same basic process. This is the central dogma that we have right here. Now, there are some exemptions. Okay, in some laboratory cases, in some laboratory environment, RNA can be reverse transcribed to become DNA. Alright? And in some cases, DNA can skip. Okay, DNA can skip the whole process to become protein immediately. Okay, the DNA can skip the whole process and directly translated to become protein in some laboratory conditions. Alright? So, but definitely, the one that you have right here, this one is the main process, or is the process that is called the central dogma. Okay? This is the central dogma of molecular genetics. Okay, so as a special part of this lesson that we have right here, so I've included in this lesson the things that you need to know about the COVID-19 vaccine. Okay, so let's check that one out. Alright, so what we have here is a bonus section of this video that we have. And since we are experiencing this COVID-19 pandemic, and lately we are... Okay, you have seen on the news the vaccination process and the use of vaccine, or the development of the vaccine versus the COVID-19. Now, very timely that this particular lesson that we have right here about the protein synthesis, it has something to do with how the vaccine was created. Okay, it follows the same it follows the process. Okay, so that's the basic process, the central dogma that we discussed in the protein synthesis. It follows, or the development of the COVID-19 vaccine follows that basic process. So let's try to find out what are these. Okay, so in a COVID-19 vaccine, alright, so in a COVID-19 vaccine, so it contains it contains parts of the virus or the virus itself, or inactivated virus, in short. Alright, so now the goal of this vaccine is to create immunity, alright, ahead of time. Okay, it's to create immunity. Now, if we take a look at the COVID virus, so the COVID virus is something like this, right? So I'm not good at illustrating the virus. Alright, so the virus itself, it contains the RNA inside. Okay, and some spike proteins over here. Okay, there are some spike proteins and the envelope. Alright, the nuclear envelope that covers the mRNA, the RNA on your virus. Okay, so what is the thing here? Now, we will follow the process of the central dogma, okay, in order to create, or in order to find out how was this vaccine developed in the first place. Alright, all of the parts of the virus that you have over here, all of these parts is made up of protein. Okay, so as we all know, proteins are made up in our cells via protein synthesis, and it follows the translation and transcription process. Okay, so that is how the molecular biologists, those who develop this vaccine, that is the basic thing that they take note of. The proteins that makes up this virus that you have right here, especially the COVID-19 one. Okay, so they develop several processes, they develop several techniques in order to create the vaccine. So there are vaccines that are mRNA, there are mRNA vaccines, and there are some vaccines that are inactivated virus. Right? So inactivated virus. Okay, activated. Sorry for the wrong spelling. Alright, activated virus. Alright, and there are some that are direct to the point. Alright, so there are nanoparticles. Okay, there are some vaccines that uses nanoparticles of the virus, or nanoparticles part of the virus. Alright, so let's try to find out how this vaccine works. Okay, so alright, so we have here the first vaccine, which is the AstraZeneca. Alright, so the AstraZeneca has an efficacy of 82% versus the usual strain of the virus, and also the US and the UK strain of the virus. And it is somehow 10% effective on South African strain of the virus. Alright, so this is the AstraZeneca uses the old method of creating a vaccine. So by the use of your inactivated virus, in this case we have the adenovirus over here. Okay, so the adenovirus contains the DNA. Okay, it contains the DNA necessary to create the spike proteins of that COVID-19. Alright, so the spike that you have over here, this is part of the COVID-19 virus. So, meaning to say that the DNA that you have inside this virus, the DNA that you have here inside this virus is not the original DNA of this adenovirus. It is the engineered ones. Okay? This is the engineered ones. And the DNA that you have over here, alright, in order for it to enter inside the cell, it must be enclosed in a virus to enter more efficiently inside the cell. Now, once it inside the cell, it will release the DNA inside that virus. So this is the DNA. Now, the DNA that you have over here is designed to create the spike proteins only, not the virus itself. Okay? So the spike proteins are one of the parts of the coronavirus. Alright? So the spike proteins for that virus. So that's the DNA that we have over here. It only creates the spike proteins. Okay? Now, once it creates the spike proteins with the use of the machinery of the cell. Okay? So if you will going to rewatch this video on how the protein synthesis work, so the DNA will be transcribed by the mRNA, and the mRNA will be translated by the ribosomes, and the ribosomes will now create these particular proteins needed to make your spike proteins that you have. Alright? So in short, your cells now has the ability to create these spike proteins. Now, once the spike proteins are in place, your body's immune system will now be triggered to create antibodies for this spike protein. Okay? So your body will now create an antibody for this spike protein that you have. So if you are expecting that you will going to produce a virus, it's not. Okay? The goal here is not to produce the virus itself, but to create parts of that virus. And fortunately enough, the molecular biologists who created this vaccine be able to isolate the DNA. They be able to isolate the genes that creates the spike protein of that virus. Alright, so it creates that spike protein. Alright, so that's the Oxford AstraZeneca. Okay, so the next one is the BioNTech Pfizer. Okay, the BioNTech Pfizer. So it uses mRNA technology. So it doesn't use, this is a new technology. Alright, this is the new technology in making vaccines right now. Alright, so what do we have right here? Instead of using DNA and an inactivated virus, the vaccine contains an enzyme that coats the mRNA, and that enzyme is very interactive with the cell membrane. Alright, so this enzyme is very interactive with the cell membrane. So once it interacts with the cell membrane, it dumps the mRNA that the capsule carries. Alright, it dumps the mRNA So once the mRNA is inside the cell, so you do now the central dogma process. Alright, so you do now the central dogma. The mRNA will be translated by the ribosome. A ribosome will approach that mRNA and it will create the proteins necessary to create the spikes of the coronavirus or the COVID-19. So the one that you have right here, these are the spikes of the COVID-19. Right? COVID-19 spikes. Okay? Not the virus itself, only the parts, which is the spikes. Okay? Again, what is this mRNA all about? Alright? What is this mRNA? The mRNA that you have right here are the segments of the virus that creates the spike protein. Again, these are the segments of the virus that creates the spike protein. Only the spike protein, not the whole virus itself. Only the spike protein. Alright, so the Pfizer that we have right here has an efficacy of 95% versus the UK and US strain. And also, it doesn't have any data at all regarding the South African strain. Okay, so now, the problem with this one is it allows or it creates the vaccine must be stored in a more lower temperature. Okay? Unlike the previous one, unlike this one, the storage temperature is only 2 to 8 degrees Celsius. Alright? So, 2 to 8 degrees Celsius. While this one requires negative 70 degrees Celsius. Okay? So, that's the disadvantage of using the mRNA technology. So, hopefully in the future, the molecular biologists will be able to address this problem in order to create a vaccine that can be stored in a normal regulated temperature. Alright, so that is the mRNA. Okay, so Moderna also uses mRNA. Okay, so it has an efficacy of 94.1% and no data for the South African strain. Okay, and in terms of storage, okay, so again, this is one of the disadvantage of this technology, the storage. Alright, so the storage is requiring negative 20 degrees Celsius. It is much higher, alright, it is much higher than the Pfizer that we have earlier. Alright, so that is the Moderna. Again, it uses the mRNA. So, if you are still confused, the mRNA segments that you have right here only creates the spike protein. It doesn't create the whole virus itself. Alright? It doesn't create the whole virus itself. So, with the use of the machinery of the cell, you will now be able to construct or you will now be able to construct these spike proteins, and these spike proteins that we have right here will trigger the immune response of your body. Okay, ahead of time. Alright, it will trigger the immune response. So when a virus enters your body, when a real virus enters your body, you will now have an immunity over that virus. Okay, so in short, you are now prepared. Okay? You are now prepared to fight that virus. Okay? Unlike what we have right now, okay, if you are not immune, your body will try to find out your body will try to figure out how to fight that virus. Okay? Now, with the use of these vaccines that we have right here, your body is already prepared in fighting that particular virus. Okay? Which is the COVID-19. Alright, so if we take a look at the Bharat Biotech vaccine that we have right here, it uses the inactivated coronavirus. Okay? It uses the COVID-19 itself, but the inactivated version. Okay? They will only they use the inactivated version of the virus. Now, what is inside the virus is very important. Alright? What is inside the virus is very important. This is okay, it contains a chemical called beta-propyolactone. Okay? It cannot replicate. Alright, so the good thing here is, since it is inactivated, it cannot replicate inside our body. Okay? It cannot replicate inside our body. So therefore, once this inactivated coronavirus is inside the body, alright, it will not enter the cells. Alright, so it will not enter the cells or something like, in this case, I think it will enter the cells. Okay, it will enter the cells, and once it enters the cells, unfortunately, it will not replicate. Okay, it will not replicate. Now, all you need to have is this inactivated virus, and once your body acknowledge this presence, acknowledge the presence of this virus, it will activate its immune response. Okay? It will activate its immune response. But since this is inactivated, it will not have any effect on your body. All right? You will not get sick, you will not get pneumonia or something like the symptoms of a normal COVID-19 virus. All right? So that's the good thing about this kind of technology, the inactivated virus. All right. All right. So the Sinopharm. Okay, so I know that you have heard this Sinopharm lately. So this is the vaccine that was used by the president to get vaccinated. So again, the Sinopharm uses inactivated virus. Okay, the coronavirus itself. This is the COVID-19 itself. Okay, so the COVID-19 virus, but inactivated version. All right. So once it enters inside the cell, it will not replicate. Okay, so you don't need to worry about this one. So once it enters inside the cell, it will not replicate. So it stays outside the cell, and it will be acknowledged by your body. It will be acknowledged the presence of this virus inside the body, and it will create an immune response. It will create an antibody, so that when a real virus enters your body, you are now prepared. Okay? So, next one. The Novavax from US. Okay, so this is different. Okay? They do not use inactivated virus. They do not use DNA. They do not use mRNA. Instead, they use the parts of the virus. Okay? They use the parts of the virus itself. So, the parts of the virus is being injected inside your body. Alright, so the problem that we have right here, according to some extent, it has somehow a lower it has a lower efficacy compared to the inactivated ones, okay, and the mRNA or the DNA supported, okay, vaccine. Okay, so the one that is injected on your body is the spike protein itself, which is attached to some nanoparticles, which is coated by the protein itself. So these spike proteins that you have right here is from the COVID-19. Alright, so it's from the COVID-19. Now, once this is present inside your body, again, the body will now create the immune response necessarily to combat this virus. So once that immune response is in there, when the real thing enters the body, you are now prepared. Okay, so somehow. Okay, so next one, that is the Novavax. Okay. Okay, so we have here the Johnson & Johnson. It uses the same technology that we have, which is the DNA. Alright, so the DNA will enter the virus will enter the cell that we have right here, and then it releases the DNA. The DNA will be transcribed by the RNA polymerase, and it creates the mRNA inside the cells. Now, once the mRNA is created inside the cells, it will trigger the machinery to create the spike proteins of your COVID-19, and once this is created, your body now will be alerted with the presence of this kind of foreign material. So your body will now be alerted, and it will create your body will now create its immune response to these spike proteins. Once the real thing enters, your body again is now prepared. Okay? So that's the thing here about this vaccine. So we have here the Sputnik 5. Alright, so the Sputnik 5 from Russia. So actually they have 2 doses. Alright, so they have 2 doses. So it contains the DNA that can support the first dose. okay, that you took. Okay, so it has an efficacy of 91% and it works the same thing as the Johnson & Johnson, the AstraZeneca. Alright, it's worked the same thing. So it uses the DNA to create the spike protein. Again, if you are confused, this DNA doesn't create the virus itself. It do not create the virus. It only creates the part of the virus. That is the spike protein. Okay? So if you are confused, if you are still confused, alright, it only creates the spike protein. Alright, so that's the Sputnik 5. Okay. So, we have here the Sinovac. Okay, the Sinovac uses also the inactivated form of the virus. Okay, so it is not designed to replicate, but it is only designed to provoke your immune system. Okay, it is only designed to provoke your immune system to create these antibodies for this virus that we have over here. All right, so those are the vaccines and that is how they are made using the central dogma that we talked about earlier on this video lesson. All right, so I hope you did get something about how these vaccines work, and probably in the future you might be the one who will discover how to create a much efficient vaccine for several diseases someday. All right, so that is the vaccines and the COVID, or rather the vaccine, the COVID-19 vaccines and the central dogma of molecular genetics. And that ends up our topic for this video lesson. I hope you'll be able to get value on the topic we discussed today. And if so, don't forget to like this video. If you have question and suggestion, write them down in the comment section below this video. It will help me a lot in the algorithm for my YouTube channel. Most importantly, don't forget to subscribe on my channel and hit the like button on the video for you to be updated for more science video like this in the future. I'll see you next time, everyone. Stay safe, enjoy learning. Peace out.
Baka みたい子供なのね